Hello, I'm Barnaly, and welcome to my presentation on early risk and late life resilience. I'd like to thank the organizers for inviting us to present some of our recent findings from the Human Connectome Project in Aging, our HCA study. From the previous presentations, you have heard about the structure and methods of the HCA study. Now, I will provide some examples of applications to answer our lifespan questions about risk and resilience. We are primarily interested in the factors that contribute to risk, for example, health factors that are known to contribute to impairment in late life, and neural or connectivity mechanisms of resilience, for example, what promotes high functioning in late life. We wanted to explore how cardiovascular and metabolic risk factors affect functional connectivity across the lifespan. To do so, we used data from 930 participants acquired in HCA to assess the impact of cardiovascular and metabolic risk on the whole brain functional connectivity using resting state fMRI data processed by the data core, which was discussed by Dr. Matt Glasser in the previous presentation. The age range for this sample is between 36 and 90 plus years, with 55.6% women in the sample. To assess cardiovascular and metabolic risk factors, we used a composite index of cardiovascular and metabolic factor, or CVM health factors, of averaged Z-transformed measures. The composite index included 12 factors encompassing vascular, metabolic, and immune systems that are known to increase risk for or correlate with CVM health. Some of the risk factors we used included blood pressure, BMI, and cholesterol. Note that higher value of CVMI indicates poor cumulative CVM health. As I have mentioned earlier, our primary goal of the study is to explore how cardiovascular and metabolic risk factors affect functional connectivity across the lifespan. Typically, when we examine the association between brain connectivity and health or any other factors, for example, in this case, cardiovascular and metabolic index, or the CVMI, we can get two different types of association, which are positive and negative associations. In positive association, the direction of relationship between connectivity and cardiovascular risk is positive, whereas in negative association, this direction of relationship is negative. But this positive or negative direction does not tell us if the correlation is getting stronger or weaker. It just tells us the direction of the change, but an effect can have a positive slope, and that can either weaken a correlation or strengthen a correlation. In addition, the whole brain connectivity effects are typically visualized as heat maps. And if we could project these whole brain effects onto the brain surface, that might be more helpful in terms of interpretation, as well as finding hotspots showing greater effects. In this study, more than 98% of the significant associations between connectivity and cardiovascular risk were weakening associations. Therefore, I will focus on explaining the conceptual background relevant to the weakening association. If the association and mean connectivity show opposite signs, then we define it as a weakening association. For example, in part A, the mean connectivity is positive while the association is negative. Similarly, in part B, the mean connectivity is negative while the association is positive. In other words, a weakening effect is when the strength of the correlation or anti-correlation is decreasing with increasing risk. We test the association between risk factors and pairwise correlation between every region and every other region within the brain. Then we sum them up as a summary measure. For the brain region, we use the Glasser 2016 parcellation as discussed in the previous presentation. The cartoon figure in A demonstrates our approach for estimating summary maps. First, we find out whether the association between CVMI and the pairwise correlation between a particular brain region with every other region is significant. If the association is significant, we assign a weight of 1 to this pairwise association, otherwise a weight of 0. For example, in this cartoon figure, 2 out of 3 associations were significant, and therefore the degree of association for this region A is 2. Finally, we sum up these weights across each region and find the summary measure, which is the degree of strengthening or weakening for that region. For corrections for multiple comparisons, we used a permutation test and applied FDR correction. Before we move on, let's take a look at an example of the weakening effect summary map. Here, the darker blue indicates lower weakening degree, whereas the light green indicates a higher weakening degree. For example, this posterior insular area showed a CVMI degree of 275, which means that in this case, when we test, there is a significant association between left posterior insular area and 275 other paired regions after regressing out cardiovascular risk measures. These are the results showing CVMI-related weakening decrease. Just to remind you, the darker blue indicates lower weakening degree, whereas a lighter green indicates a higher weakening degree. First, we looked into the whole brain weakening degree without any adjustment for age. These results show a global weakening effect, which is summarizing the total number of connections across each brain region that showed a decrease in connectivity with increasing risk. After adjusting for age, while the total number of significant associations decreased, we can still see similar patterns of weakening across the brain, showing a large effect. We also found strong bilateral symmetry in weakening effects with and without adjusting for age. Some notable regions with more weakening decrease included regions in the frontal and lateral parietal cortex, as well as several regions in the medial prefrontal and cingulate cortex. Note that any regions showing a color in this map are regions that showed a significance after permutation testing and FDR correction. Also, I'd like to note that in this context, strengthening or weakening does not necessarily indicate functional integrity. We also wanted to examine how these weakening effects are distributed across different age groups. For example, we wanted to know if the effects are replicated across middle-aged and older groups, or if one group is more affected than the other group. The younger middle-aged group is defined using the participants below 65 years, while the older group is defined using the participants who are 65 years and above. These are the weakening effects for younger middle-aged as well as the older groups. From these figures, you can see that while the effects are fairly replicated across the brain, the younger middle-aged group is showing relatively greater weakening effect compared to the older group. Also, the older group is showing greater weakening effect in the primary sensory motor areas, whereas the younger middle-aged group is showing greater weakening effect in the limbic region. Finally, we also wanted to explore how weakening effects appear in the 80 plus or the oldest old sample. While we had a much smaller sample for this group, we can still see some patterns of weakening in the temporal lobe, similar to the other age groups. We also used a cognitive composite index to first identify high and low performers based on the top and bottom 50% of the sample across the age regression line. We found that overall, the low performers have higher cardiovascular risk than the high performers across the age span. In future, we will look at how brain connectivity, cognition, and risk all are related together. In summary, cardiovascular and metabolic risk effects are stronger in younger middle-aged adults compared to the older adults, which highlights the need for early attention to cardiovascular and metabolic factors in adults for optimal brain health and functioning. This work would not be possible without the contributions from these individuals listed on the slide. Additionally, we would also like to acknowledge the support from National Institutes of Health, including National Institute on Aging for the current study. Next, Dr. Melissa Terpestre will give the final presentation in which she will describe novel magnetic resonance spectroscopy applications to the Human Connectome Project Aging. Thank you all for listening to this presentation.